sunrise brings a new day filled with new hopes for a new beginning. A very good morning. Here is another day of learning for our inquiring mind. I would like to welcome Dr. Nagaraj Nagatehalli, Assistant Director for Basic Science Research and Associate Professor of Surgery, Sylvester Pancreatic Cancer Research Institute, University of Miami, USA. And I would like to welcome Professor Chandrasekhar G. Joshi, Coordinator MSc Biochemistry Program and Coordinator GAN Program. And I would like to welcome faculty members of Department of Biochemistry Program. And I would like to welcome our enthusiastic participants. Have a good day.
Thank you. Welcome all. I think hope you had a fantastic day yesterday. Learned something. Today, so we're gonna have some journey. Okay, this is I think important. That today's journey, I hope you enjoy it. It's important because whenever we say we have to do research or on cancer biology, then everyone will have some doubt, okay, where do I start, right? How do we do it? You might know technology, you might know in vitro, in vivo, all this methodology. You might have heard somewhere, you might have seen someone doing it, some lot of papers published, and either you're a biochemist or the biotechnologist or the genetist or any subject that you might have done in a master's or during PhD program. But when you want to start some research or any program, doesn't matter if it's a cancer biology or any other, you have to start from somewhere. You just don't know where to start, right? So there's the same situation I had it too many years, right? We have to start somewhere, right? How did I start it? And then how did I go ahead and discovered many things that you're going to see today, either as a researcher, as a publication, and also from the publication, how did I do the clinical trials? How did I accomplish any bigger paper? And what it took for me to establish in the whole program of the pancreatic cancer, you will see the journey. So it's very important as a, if you want to do research or if you want to accomplish something in the cancer biology. So I think this story probably will help you to develop some concept and ideas so that you can actually do something your own in your laboratory or your university or institutions. So this is called the translation journey from the molecular mechanism to early phase clinical trials. So whenever we talk about the cancer signaling, right, we talk about normal cell and the cancer cell, right? Normal cell, you all know, functions normally. Cancer cell, when we say cancer, your question will be, the first question will be, what is the big difference? You always have, as a classroom, you might have learned what is cancer cell, what is the difference between normal cell and cancer cell. In the research field, you have to understand that when you have a cancer cell, that means that there is a fermentation. There is a fermentation process in cancer cell. That's why the cancer cells have this less oxygen, more CO2, right? There is a less oxygen in that's the environment is that created like in a fermentation environment. That's why these cancer cells needed that. They needed a lot of sugar, right? And also energy, right? They need energy. So they basically, the mitochondria and the cell with the fermentation process, that means that glutamine, you all learned the glutamine, and they need glutamine, right? So there is a high glutamine. So in that environment, so that's why it's cancer cells divide and become cancer cell, right? And also there are a lot of other functions I can talk about, the different big difference. But important, I think, the fermentation process that takes care within that environment. Sugar is bad for cancer cell. That's why we always say diabetic patients, you have to be careful. They're very sensitive to become, they're sensitive to the cancer. The reason is that their body is like a fermentation process, like there are a lot of sugar in the body. the cancer cell loves sugar, right? And also, they don't have energy. They have less energy. That's why not all the sugar patients or the diabetic patient will not get cancer. They have less energy, and they need a lot of energy. Mitochondria is another energy source, is also important. Healthy mitochondria we have to maintain, right? And if anything you want to do, conduct research on mitochondria, sugar, and metabolic process, or the glutamine or the fermentation process is a very important research area where you want to, people also say, probably some researchers might know, normoxia, hypoxia, and all those studies, right? So one of the common factor in any cancer, doesn't matter, I've mentioned a couple of cancers here, that doesn't matter which cancer you take, the common factor is a fermentation process that takes care, that will happen in any cancer, doesn't matter. There are a lot of things that we can talk, like, different cancer have a different mutation and different, way of, process in the progression process and the metastasis process, but the common factor is a fermentation process, right, and a metabolic process. So that's one thing that you need to remember, that if you conduct that kind of a fermentation, whatever that I'm talking about, hypoxia kind of a study, that's one process that you can actually commonly do in any cancer. It's the same thing. So if you build some area around that, what I'm talking about, okay, you can listen to a lot of researchers that are working in cancer biology, but it's the same thing though. They're going to talk about this. But what they're not going to tell you is that the common factor that you can design as a researcher, that then you can start studying a colon cancer, breast cancer, prostate cancer, brain cancer, anything cancer you take, but your one process that you do in the laboratory is the same thing, doesn't matter what cell line you're going to do. Like that. For example, people start with the SARC signaling. Like I started with the SARC signaling. SARC signaling is common in many signaling cancer cells. It happens. Okay, that's one of the signaling. I'm going to tell you why I started with the SARC, and then we go ahead with whatever the process that I've done in the research. But that's also important. It's the same way. Many investigators start with different signaling pathway. So I started with the SARC signaling. And then I went ahead and did a lot of other different studies. But I'm going to explain to you, why I did different studies. There is a reason for everything, right? When you do one research, one particular topic, right, that should link to the next topic that you're going to conduct, and the next, and the next. Then only you establish one area which is yours forever. So this is mine forever. So it is like that. So we published, I published as a first author, I think, 4 or 5 papers from this study, old study. And then I became a senior author, and I started working towards that. And then I left and I started another investigation, developed my own area, my own research. But they all linked somewhere, somehow. But the linking process is there, right? That's what I think you need to think all the time, that you need to link each study one another, and you should not, it's easy to start another project, as I said yesterday, but it is difficult to continue the work that you're doing every day, right? So this story is about from SARC, EGFR to STAT3 and the ERK, and then a lot many things, okay? This is one signaling pathway. This is a tumor intrinsic study. After this, I'm going to talk about tumor microenvironment, like yesterday, but differently, right? But this whole process is a tumor intrinsic study. For example, the whole study that I have done, right? I have done in the pancreatic cancer, but people have done afterwards. Doesn't matter which cancer cell you're going to take. It's the same signaling pathway they have published. There are tons of papers that have been published. Okay, that's why it's very important. You become the first, and then people cite your paper for a long time. If you don't become a first person, then you don't get the citations much, right? You become first all the time. But that study has to be unique. That study has to be new. That study has to have some result that actually helps patient or something in the future, or the trial or something else, right? Or maybe that is very important discovery, right? That is the story. So you all know that there are many reasons for therapeutic resistance and lack of progress seen in survival outcomes, just not in the pancreatic cancer, in many cancers too. So there is the resistance pathway, therapeutic resistance was the biggest problem in the pancreatic cancer during the time that I started my research, right? There's a heart tumor, I talked about heart tumor and there was no drug delivery. These are all the problem. I took one problem that is a resistance pathway problem. So that resistance, you start somewhere though. You have to start the research somewhere that you take that biggest problem. For example, lung cancer may have another problem. What is the major problem with the lung cancer? Smoking. And smoking is the biggest problem. But now there are no smokers, not much when compared to 30 years ago. That's why the lung cancer burden has decreased comparatively. Just imagine if they have not done the anti-smoking advertisement or all these things, anti-smoking, whatever the labels that you're going to see, if they have not done it, probably the lung cancer patient probably will be very high. That there is something that government has done and they keep doing in worldwide. That's why the lung cancer is going down. But what I'm saying is that you take that whatever that cancer that you wanted to study, but it's important to look at the problem that what that cancer has, the biggest problem. Take the biggest problem and start looking into the signaling pathway or anything that you want to study. For example, I'm talking about signaling pathway because I'm a cellular signaling person. But you can think about anything else. Think about metabolic pathway. Think about something else that you wanted to study, or hypoxia, or something else, or maybe transcription regulators. You talk about only the transcription or epigenetics. You start with epigenetics, it's fine. But I'm just taking one problem I'm trying to establish. The same way you can establish in any problem you take, based on that what experience that you have, what background you have. Okay, I had a background biochemistry. I had a background some signaling, enzymology, and studying kind of biochemical pathways and all those. I was always interested in cellular signaling pathway because I get attached to and attracted towards it. But I've also done something molecular biology. So I've kind of linked all these studies, what I have experienced during my master's and the PhD program. So that you can think about your background and think what you can do and how you can establish based on your experience, okay? And the problem that I took when I started the lab, I also told you that I met a clinician and then I started the lab. That's what I'm talking about. So I started with one problem, that therapeutic resistance had a bigger problem in pancreatic cancer. I started with that and then started studying what are the challenges to new drug development. I started studying, looking at the targeting a single pathway whether will be successful or not. So already there were people who were talking about, oh, targeting single pathway may not be successful. I had that in my mind. So you have to know that whether targeting single path, but you have to start with somewhere though. You cannot start with everything together one time. You have to start somewhere, right? I started with single pathway only. I want to recreate the thing that people already started saying, targeting single pathway is not enough. Okay, fine. I want to see that too, because I want to experience myself that it is not enough, right? I want to prove again that what others have proved. Then I will also learn during that process what to do, and that's why I started. So I started with single pathway targeting, right? And then I also learned during that I did a lot of literature survey, and then I also learned that the presence of dense desmoplastic stroma. I didn't know about pancreatic cancer at all at that time. If you see this picture, there is a desmoplasia overall. There are a lot of, this adenocarcinoma here. That means that it's a cancer cell here. But not many cancer cells around in the pancreatic cancer, but there are a lot of desmoplasia, means stroma. I knew that. I saw these pictures everywhere, and these kind of pictures everywhere they were running during that time. Okay. So I started understanding, characterizing the fibrotic tissue. What does that mean? Vasculature, function, all these things. And also, I learned that during that time, incomplete understanding of the biology of this disease. That's what I think I was excited about. Okay, they were saying that, oh, there is an incomplete understanding of the biology in this cancer. At that time, there were not many investigators working all over the world, and even in the US, on the pancreatic cancer. I thought that's very important. When somebody is a commentary they're writing in the journal, they're talking about, okay, what is the future? What do we need? I think that's a very important thing, that when you are studying anything cancer related, you look at the commentary from the expert. They talk about, okay, this is what is lacking in this field. It's a key factor. That's what I think you need to look into. And then you start thinking about, okay, why are they saying? Why are they saying that, this is important, you should develop? Means that they're not, it is not that they're not able to do it. They want next generation to take up the problem and do it. That's you, right? You have to take those problems and investigate. It is not that they're actually giving you a hint what is important in this field. That hint is actually is an important thing. That's how I also got into that, understanding the biology of this disease is incomplete. So then I started looking at the mechanisms of interaction of different signaling pathways, and what do you mean by tumor microenvironment? At that time, tumor microenvironment, when 2007 and 8, it's very low. Not many people were working on the tumor microenvironment. But for me, the attraction was the signaling pathways, right? Because I was always, wanted to study those things. So to start with, there were investigators around Vanderbilt University. I told you this is in Harvard University. It's one of the top university in US. So it's that when I joined Vanderbilt, so there were a lot of investigators in the university. They were the investigator, scientists who actually discovered SARC. They were there, and there were scientists. They were actually working on the STAT3 signaling. There were scientists working on the JAG, the EGFR signaling. There is a whole group working on the EGFR. They spent their lifetime on the EGFR signaling pathway. And there was a guy who actually investigator and also scientist who made a discovery in many signaling pathways. And also there were discoveries for GPCR, and there was EGFR. The whole team of biochemistry in Vanderbilt was discovered EGFR, many different pathways during in the sub pathways. So I thought I should start somewhere around that, because all scientists are around. If I want to have discussion with any of these investigators, they're all there. They're all in the coffee shop. They're all working for, they go for lunch. I can go to their laboratory right across, and there were postdocs and the graduate students running around here and there, so I can even talk to them and see what they are doing in their laboratory so that I can learn. This is very important, that interaction, going to the conferences and going to symposia, attending and meeting other people, talking to them, and looking at their data and the papers is a key factor. That's exactly what I've done. So I looked at the surrounding and environment. Okay, this is where I'm going to start. So if I fail also, they will tell me why I failed. If I pass also, they will tell me whether you're right or not, right? So that's why I started SARC, EGFR, JAK, STAT3 signaling pathway. But my first thing, I started with SARC because there was a pioneer worker, scientist and a discoverer right next to my lab who actually worked on SARC. Okay, that's why I started with SARC. I thought, because no one worked on pancreas around, they all worked on one signaling pathway. They don't specifically, when they discover the EGFR or SARC signaling or whatever, they don't discover in this cancer or that cancer. They discover, but it's you to take those discovery to your interest, whichever cancer you want to work on, right? So that's important. Like ERP pathway and this, many pathways that they all discover, but they don't know. They don't tell you, where you want to take this one and work on. That you have to think about it. You have to read those papers and understand the paper and see whether you can actually work on those. Okay. So I started with SARC, and the SARC, can be, attract many signaling pathways, SARC FAC, SARC STAT3, and all those things. I learned all these things. There were a lot of literature. There was less literature, but there is enough literature to understand the whole, the combination of these pathways. Okay, and this is very important as an investigator, that understand there are a lot of signaling pathways, linking each other, right? just not one. Like for example, if you take different cancer, and there are a lot of signaling pathway cancer. There are like 33 cancer types here. There is one paper, nice paper actually. You should read. There is like 9,000 around tumor samples they have taken, analyzed that signaling pathway. There are like tons of signaling pathways you can actually look into. When I started, there was nothing. But literature is an important thing, and that too, reading a right literature and a good literature is an important thing. For example, if you don't have access to one journal or paper, you can always request to the senior author or someone who actually can find an email address and look at it and then ask them. They will send you. If they don't send you, remember, everyone is busy, okay? So that you send again and again and again, they will send you back, okay, one day. So, but if you want to see any literature, and please request to the senior author, and they will send you. If they don't send you, there is always a way that you can find, how to find. There are always friends somewhere around, they can actually find for you. That's important. Reading a literature, key thing, right? For example, if someone would have published like this before when I started, that would have been fantastic. Probably I would have saved a lot of many years, right? But now you have it. But you just have to find out what you want to study and look at it, look at those signaling pathways. I think that this is where I started, not this exactly, but what I'm saying is that I started with from the scratch. But you have something that you can look into this literature and you can find what you want to study. And also there is a lot of signaling pathways and resources available. If you go to that Cell paper, there is a lot of signaling resources that you can actually look into those signaling pathways and their laboratory website, and also there are a lot of public websites that actually you can look into. There are a lot of resources available. It's easy. It's just that what they don't have is that they don't know what you wanted to study. So that's why they're giving you information so that you can actually look into it and study. So this is something that I didn't have it, right, when I started. So when SARC can interact with many GPCRs, I knew that. It's like, many receptor tyrosine kinases, example, MET, PDGF, R, EGFR, and integrins, and this kind of literature I had it, kind of understood that whole signaling. And also SARC can phosphorylate P120 catenin. The information, the P120 catenin was investigator right there who discovered in Vanderbilt. And so it is from their paper though that they talked about stabilizing the E-cadherin and all those things, right? I actually wrote papers related to that in pancreatic cancer. Remember, I started pancreatic cancer lab, so I started looking into the same signaling pathway they were all working on, and I took all that information and I studied in pancreatic cancer and published a couple of papers on it. So that's how that's how you should do it too. You can actually look at it, who actually discovered what, and see their paper and read about what they have done, and then you actually look at it in your own model and see what you can actually contribute in that pathway. And also I looked at what SARC do other than just the SARC itself. And I kind of understood that SARC and FAC and a lot of Paxilin and ECM factors and the Rho kinase and a lot of these things you will hear from me again, Rho and ECM and all. Even yesterday I was presenting about rock signaling. So this is kind of linking everything in my research, like whatever I was actually finding out 15 years ago. But they're all not just I'm linking, it's just happening in my research also. But whoever discovered these things, they have discovered right. That's why I was able to get the right pathways, that even though in the model is different, that mine is pancreas, and nobody has worked on the pancreas, in these signaling pathways, other than what I have done. What I'm saying is that they all link. This paper is from 2023, okay? This is recent, okay? They're talking about the same thing that I'm talking about, my work during 2010 or 8 or 7, right? So it's important, right? Then what I did was understanding that SARC kinases in pancreatic cancer. That's how I started. So then I started working on the SARC. What is SARC? And then I ordered an antibody and then cell lines from ATCC, and then I pancreatic cancer cell lines and the normal So I looked at the normal cell line. Nobody had it at that time. And then later I got it. Things like that. But somewhere I have to start. I started with SARC, okay, because of all these reasons. Then what I did, I didn't know what to do, okay. What I did, okay, how do you start? I'm a therapeutic person and also signaling person. That's what my experience is. What I did was I ordered dasatinib. That's the only drug available at that time. That is a dasatinib. It was actually clinically approved at that time. So I ordered the dasatinib drug. I ordered a couple of cell lines. I started screening. Okay, BXPC3, PANC1, these are all pancreatic cell lines. I started screening with different concentration, different time point, and then I saw that it is signaling. This signaling is inhibiting. See, all these kinases I was actually talking about, I actually showed you in other pathway. They're all linked. I only ordered those antibodies which actually linked around the SARC, and then I started looking into it. And then, what I found out? There was a sensitive cell line. I think yesterday you were talking about sensitivity and non-sensitive, resistant cell line. This is one way. You have to look at it. Different cell lines also have some cell lines sensitive, some cell lines are not sensitive, resistant. For example, here BXPC3 was sensitive to this, and it inhibited a lot of STAT3 and MAP kinase signaling, but it did not inhibit in the PANC1. But it is a resistant cell line to this signaling, even though the drug is same. It is one patient here, pancreatic cancer, another patient, pancreatic cancer. It is not that one, same drug will be can be used for all the patients from the pancreatic cancer. No, it may not be effective. That's why we will not be able to survive every patient, right? The even patient have one patient to another patient. There's a big difference there. Example here, right? In this signaling pathway, we were not inhibited in the resistant. I got attracted to this. That why? How do I come up with something logic like yesterday you were talking about? The same thing I was actually thinking about it 15, 20 years ago. So then I thought, okay, this is something interesting. Then let me look at it. What's going on here? Then my finding I don't want to go in detail in the SARC signaling, what I did and all. I'm going to tell you what happened and what exactly, how I overcome this resistance pathway. So SARC inhibition resulted in decrease in cell proliferation. I looked at, you all talked about what did you do, what do you do when you take something to study, cell proliferation, migration, invasion. I was talking about the anchorage independent assay, colony formation assay. These are all the common signaling pathway. Even during that time, even today, people work on those things, but people don't show a lot of this data. They put it in the supplementary data here and there, but they'll take the question, the bigger question they have in each paper, and they work on it. But these factors, just they're screening factor for them. But they may be investigating something huge. That's why they go into that particular aspects. That's why they don't show everything. But for you, probably that may be not enough resources to go there, but you want to show something what happened in basics, it's fine. But it's all researched data, so you can actually present whatever you want, that is convenient to you. So, and also I saw reduced phosphorylation of key signaling pathways, say proteins like FAC, AKT, STAT3, ERK, which was observed. And in vivo, I actually used the dasatinib treatment in the tumor growth xenograft models and all. So I'm not showing all those papers, it's all published long ago. But what I'm saying is that I did all the studies, but my curiosity was that STAT3 signaling, MAP kinase signaling was not inhibited in the resistant cell line. That is the curiosity. But I did all those things, and I published that paper too, okay? That paper was published, just a SARC signaling and resistant pathway, that's it. It's over, done. And then I was actually looking at it, okay, what do I do now? How do I overcome this study? Then I did a lot of different studies, a lot of different drugs, and then only thing that you're actually seeing it, what I have done to overcome, right? And there are a lot of other things that I've done that actually screened but did not work. I was thinking this, I was thinking that, but it did not work. The one it worked, actually, I'm showing you. So what I did was I combined SARC and EGFR. I'll tell you why I combined SARC and EGFR. So to overcome this limitation of the single agent SARC inhibition by combining the SARC inhibition with EGFR, and also I added the gemcitabine chemotherapy, okay? Because gemcitabine chemotherapy, at that time, gemcitabine was a standard chemotherapy. Now gemcitabine, and also you have to have a paclitaxel. At that time, it's only gemcitabine. That's why I added the gemcitabine chemotherapy to that, and I studied the whole signaling pathway. Okay, and also I found out many other cell lines, just like what I showed you BXPC3 PANC1. I selected a lot of different cell lines. One cell line, two cell lines is not enough. You have to have, sensitive cell line, couple of them, resistant cell line, couple of them, so that you have to select those resistant and sensitive one and then work on. I have done all those screening, many cell lines, and I selected the best sensitive and the best resistant cell line. I worked on all the signaling pathways again, FAC, AKT, ERG, Jun kinase, STAT3 and all those things. Then I did also the cooperative inhibition of the same thing what I did in the SARC paper. And then here, again, I did the migration, invasion, colony formation, tumor growth, xenograft assay, and all those things I've done in vitro, in vivo, and everything. And I was able to show that when you add all this triple combination, I was able to overcome. Remember, during that time, this itself is huge. Today, probably if I do this, people laugh, but that's how the research is, because it's evolved. This last 15 years, it's evolved. But the concept is this, the concept and the design, that research you do, it may be very simple there, but it did work. That's, I think, important. And then I did in the in vivo tumor studies. I took the tumors and also looked at the signaling pathway, which is actually STAT3. STAT3 was actually resistant pathway, but here it became sensitive pathway. STAT3 has become sensitive, even the resistant cell line in the xenograft studies. I think that is the key important signaling pathway I found out, that is a STAT3 signaling. STAT3 signaling, why? Because in the resistant, in the dasatinib, that is a SARC inhibitor or the EGFR inhibitor I have used, and for that, sensitive means that the STAT3 became sensitive signaling. That means that STAT3 is important. Means that in the sensitive cell line also, in the resistant cell line also, STAT3 I was able to overcome with combination of SARC and EGFR inhibitor. But now if you go look at it, SARC, EGFR, STAT3, there are many papers been published in different cancer. That means that it is same problem in any other cancer. It's the same STAT3 if you use it. Then inhibitor, STAT3 inhibitor, probably you can sensitize many other signaling pathways, okay? And then I went ahead and did the SARC and EGFR. I took the sensitive one and the resistant one. I did a lot of in vivo studies. That's most important thing is the combination of SARC, EGFR, and the gemcitabine. That is a combination that I was really excited about, because I published in Clinical Cancer Research. That's one of the great paper. After that, there were some clinicians who were interested. They were like medical oncologists who were interested. In Vanderbilt University, there was a medical oncologist who was actually want to run the clinical trial on this, because at that time there was no cure for pancreatic cancer. There was no studies in pancreatic cancer in vivo therapeutically. We were very lacking. So wherever the medical oncologists will see some good result, they go ahead and do the trial. That's how the field was at that time. But today is different. But I will tell you the story afterwards, why it is different, because the science is evolved. During that time, that itself is more than enough, because even there were no investigators to investigate even that much during that time, because so that there was no hope for pancreatic cancer at that time. This medical oncologist came to us and wanted to study this in the patient. Then we were like very happy, because there's some translation. If that works in the patient, that will be even better. That's what we thought. Then they went ahead and did the phase 1 trial on this, and there were like 19 patients. They treated with the SARC, EGFR, and gemcitabine at that time, and there were like great result. There was safety, tolerability they studied. There were some preliminary efficacy data. There were a lot of biomarker analysis they have done, and this study also established the feasibility of targeting the multiple tyrosine kinases in a combination regimen. Means that, remember, in the first slide I showed you, single pathway is not enough. They were right. Even I went ahead and studied the SARC signaling, single pathway. See where I went. I went to saw EGFR and combined, and then went to the STAT3 signaling, which is a marker for me. But I went back again. I'm actually telling them again, yes, targeting multiple pathways is important. I'm coming up with the same logic what they have told me like 3 years ago at that time, right? It's right, but important is that even though I went ahead and did the SARC, I ended up in a clinical trial, right? That's what they were giving me a hint that multiple pathway targeting is important. So, because I cannot start multiple pathway, I didn't know where to start. So you have to start somewhere. So I ended up with, good result. Probably it was helping many patients. I think the patient probably survived a little longer than what it was at that time. Probably they were able to see the and the patient were able to see the neighbors and the friends and the family for a little more time, right? That's, I think, was a key important step for the pancreatic cancer at that time. So now what we found out at that time was, I was curious, this is actually published in 2020, there was a 10-year gap, right? There was a 10-year gap between 2010 to 2020. But I was always thinking about this, how this is working. That question always lies to you in your mind, right? So you know that you threw the drug and worked everything fine, fantastic, right? And then someone came and took this study to the clinical trial, and they are actually giving this drug to the patients. Fantastic, great. But as a scientist, I always will be thinking, how this is working? First of all, how this is working? Can someone show me how this is working? It was always I was thinking, how this is working, right? So, what is the technology to see whether this, because everybody was saying this is a heart tumor, drug will not go to the pancreatic cancer, even though you treat, doesn't matter what, and all those things. Then how come the drug is going, right? Then one day, I was walking by, and there was a bio department in there. They were actually doing a mass spec analysis. So what they were doing, they were taking the tissue, they're putting the drug and see whether the drug is going inside. Rather than that, if I give because I'm already giving the drug to the mice, if the drug is going to the patient, can you please analyze this? The drug is going inside the tumor? They said, yeah, that's easy. We can do it. I was like, fantastic. Wow. Then I can see visually whether the drug is going or not. Then the next question will be, if that is going, how it is going, right? Okay, first of all, let's see whether it's going. So what we did was we did the combination of all these things, and we took this, mice study, treatment study. DTB is a dasatinib, ERL is a erlotinib, that is a EGFR inhibitor, and the GEM is a gemcitabine, that is a combination that we did the trial. And then you see combining all those, we actually see the increase in the survival. Remember, this mice, we did not study at that time. We didn't have the genetic mice model, okay? They were later on, this genetic mice model was developed by another investigator in the Vanderbilt University. We came to know, this investigator investigated on this genetic mouse, which actually pancreatic cancer specific. We didn't know around. Then I came to know they published the Genesis paper. It's a great paper. And then we asked them, okay, can we use this? This is a pancreas model, which not even a single person published any papers on the therapeutic in the field. Then this is the first paper that we published on this mice using the genetic mouse model, which actually naturally, spontaneously develop pancreatic cancer, which is a great model. If I can treat these mice with these drugs and see, that is fantastic, right? So that's what we did. We treated these mice and we saw survival so good. That's why the trial is working. Because trial was done in 2010, 11, 12. Now I'm studying now in 2020, same thing in a different model, but survival was increased so good. That's why the trial was good. That's how the medical oncologists came and studied. Probably they knew it might work. That's because my curiosity, always that curiosity actually dragged me to understand and find out. Doesn't matter if you see some model later on after 5 years, and if you want to study the same study that what you have done 10 years ago, you should go ahead and do it, because that is science, right? You can understand even better, and that is actually rediscovering your own study, right? So if you see here, this is a mass spec analysis. We just did the random, scanning of all those. All the tissues that are pancreas tissues we have taken, and we just did the mass spec analysis to see whether dasatinib or erlotinib or the gemcitabine going inside the tumor or not, okay? These three tumors exactly is a random study. These three tumors are exactly the combination study. Okay, that means the single dose, either the dasatinib or the EARL or EARL or GEM or the dasatinib or the EARL or anything did not work at all. Even though you take the gemcitabine alone, like in a chemotherapy alone, it did not work. Okay, that's why the patients were dying even faster. They were like 5 months only, even gemcitabine treated. And even though SARC, EGFR, whatever that you treat, it's not working. Only combination worked is the triple combination. When you have a SARC inhibitor, EGFR inhibitor, and gemcitabine chemotherapy together. That's why I was able to see the survival so better. And in this mice model, spontaneous mice model, genetic model, and now I see the tumor had this drug, gemcitabine delivered. That means that when you inhibit SARC and EGFR, add gemcitabine to it, the gemcitabine is going here to the tumor. That means that is why, because the curiosity again, how? You may ask, you inhibit SARC, you inhibit EGFR, now you add the chemotherapy to these patients and the chemotherapy is going to the tumor, then it is killing the tumor. That's why these patients are survived better, right? Now the question will be, how? Yes, we're going to discover next. Why, how, and also. That's the research. That's what I'm trying to tell you. That question, how, why, what, how, all these things you have to have all the time in the field. Then only you will be able to establish the next paper and next investigation, probably next another trial, maybe better one. Probably today, these patients survived maybe 8 months and 9 months. Probably if you keep investigating, probably they can survive even better. Maybe 1.5 years, maybe 2 years, maybe 3 years. So that is an investigation that we need to look into, right? So what I did at that time was, okay, STAT3 is important. We thought that's one of the biomarkers for therapeutic resistance in pancreatic cancer at that time. Then my curiosity, one thing, I put that SARC and EGFR aside, and I looked at the STAT3 more and more, importantly because the SARC, EGFR, when I inhibit, I'm inhibiting the STAT3 signaling. That is curious. I was curious more on STAT3 signaling than the SARC and EGFR. Maybe it's all about the timing. You never know why, because I will tell you the story why, okay? Because it's all about when you are reading papers, research papers or journal paper, interacting, going to the conferences, meeting all these posters. You see the poster and there are a lot of new investigations and all. At that time you will see something, it will fetch in your mind that probably there is something going on here. That's exactly what happened. The STAT3 fetched me during that, I went to conference, AACR, American Association of Cancer Research, that's one of the big conference. And worldwide, when compared to a lot of cancer researchers go there. So when I went there, I was just STAT3. There were a lot of people started working on STAT3. So at that time, it is actually I'm talking about 2010 or after 2010-11, around that time. Then I was actually we worked on we wrote a grant and we got the grant on the STAT3, just the STAT3 signaling in pancreatic cancer. That's it. It's very important. Okay, we got the grant 2011 exactly during that time, because that is a big grant, right? That was like, we were so interested, and also the reviewers also see the same way, because any reviewers will see there is something new coming up, and it may be something like, important, and the translational, probably it's going to help the patient. They're going to give you the money to study that type of study. When they don't give the, why they don't give the money? Because they don't see any thing that coming out of your grant. Sometimes you end up in probably nothing, right? They can easily, reviewers can see, what's going on in this work, whether it is important or not to the field or not. That's how the novelty, innovation, they have to see it. So then during that time, in 2011, all these 3 papers came out. Look at this, what happened. So it actually came out in April. We published in around February, March. We didn't know this paper at all. They were actually in the background, the people were working on STAT3 signaling. All this paper 3 papers on pancreatic cancer. We didn't know. We were actually working on the same thing. We didn't know. Look at that. We got the grant, we got the papers and everything, and someone published 3 papers on STAT3 signaling, right? If I was like, 1 year, late, I would have not got the grant at all. Because the papers are out, there is no innovation. Because already people knew. It's just about the timing. Sometimes you have to leave everything and you work on because it's important. Important for your career growth, important for your lab, important for your future. Also, so sometimes you will think immediately, I think this is important. Yes, it is. If it is important, leave everything. Don't go to the marriages, don't go anywhere. Just work on in the laboratory. That's how these scientists are, right? They work day and night because someone else will discover the same thing. If that is something new, something exactly happened to us. Just look at that cancer cell paper. There's the big papers in the field, right? Cancer cell is a big paper, high impact papers. They came all 3 in one journal, one volume, all 3 together. They were all working together. All these scientists all over the world working together. They combined all these 3 papers and published it. It's about timing, right? The research is always about timing. If you think today it's important, I have to go back work, please go back and work. This is important. If you see something that is going to happen. So that's exactly, I'm just giving you a hint why that is important. That timing in research is very important, right? Don't delay. If you want to do something, keep doing, you'll definitely see it. So so what I wanted to say here is that all 3 are related to STAT3 signaling in pancreatic cancer, okay? they are working on different way, but end of the day, it's all about the tumor progression, metastasis, and all those things, right? But the signaling pathways are related to tumor microenvironment. There is maybe other signaling, but they're all related to one signaling pathway, that is STAT3, okay? That's important. So then I thought, okay, what is the difference between their paper and my work? My work was on the therapeutic resistance overcoming, and therapeutically much better than what they are doing. Even though they have published an high impact paper, therapeutically they are not talking about anything. They're only saying that STAT3 is important, signaling pathway. That is science. But for me, how do I treat my patients? What do I do next to investigate? Doesn't matter what people publish, high impact paper, doesn't matter what it is. It's important is that whatever you are thinking is always you are thinking right. That's actually another example I wanted to prove and show you. Okay, always I was started thinking, okay, even though they published that high impact paper, I know that people might not take my paper the next time when I say STAT3, but still I wanted to continue because my idea is entirely different than what they have published. So I went ahead and targeted STAT3, okay, and started looking at the STAT3, okay, more and more, and I started working on different cells from the same mice model. There is a KPC mice model, there is a KRAS P53, remember I showed you yesterday? That's the same mice model, but I got the cell line from another investigator from San Diego, is California. They had this line from the PANIN, that is an early neoplasia line, and there is a PDM, is a pancreatic ductal adenocarcinoma, that is called cancer cell line, and this is a liver metastatic cell line. That means that when the cell line is very aggressive and metastatic, then that we can actually derive that cell line and make it like a metastatic line. And when I saw that what happened to STAT3, look at this, what happened. So when PANIN, there is no STAT3, but when it becomes cancer, there is a high STAT3, and when it is metastasis, there is even more STAT3 expression here. Then that was very important for me, right? Look at that. In the progression of the cancer, STAT3 is increasing, right? That's the important thing. That's from the mouse line. And then I took the human pancreatic cancer tissues and analyzed all the tumor tissue microarray at that time. The result 2010, 11, 12 during that time, after the paper got published there. And then I was working on this one, on the different progression of the cancer in the human tissues. Then I analyzed all the survival analysis, STAT3, if it is STAT3 high, what happens? When you have a STAT3 high, there is a worse survival and all those things. And I got all those data. And then also what I did not show is that when you have a STAT3 low and there is a high phosphokinase, I'll come to that later. But important at that time for me is STAT3. STAT3. So what happens to STAT3? How this is happening and what is happening in STAT3? Then I targeted and did with the AZD1480, that is a drug from the AstraZeneca. And then we saw, this is in vivo data. This is hard to get the in vivo tissue analyzed for phospho-STAT3. So then we saw that STAT3 was down, and with the gemcitabine, there's a chemotherapy. It did not, nothing has happened. And when you combine with the gemcitabine plus STAT3 inhibitor, also STAT3 went down. Very important, okay? If you want to treat patient, right, I'll do something like, big study. So make sure whenever the combination you combine, your signaling pathway that you wanted to study, you're still inhibiting it or you're not reactivating it. For example, you add a gemcitabine and probably your STAT3 will come up, maybe because of the different signaling pathways affecting the signaling pathway, because STAT3 is inflammatory mediated. That's why you need to check that and make sure that your STAT3 is still down, okay? It may be important however you do, you have to think logically and keep doing the same thing and again. But at that time, there was a paper on SPARC. It is a marker for the stroma. So SPARC is one of the markers. So there was a paper came out during that time. I wanted to see what happens to stroma, because stroma is a big factor, right, in pancreatic cancer. I want to see just inhibiting just a STAT3, whether I'm doing anything to the stroma. That's why the marker I checked, the marker went down. Means that it is STAT3 is affecting the stroma. Probably our SARC and EGFR inhibitor, when you inhibit and add gemcitabine went into the tumor with a mass spec analysis, probably it is inhibiting STAT3. That's why the SPARC is going down. That means stroma is reduced, or something happened. That's why the drug is going to the tumor. That's how I was thinking at that time. Then I thought, wow, that is maybe what I was thinking, maybe right. Look at that. I'm thinking there's a paper, it doesn't matter what they published, Nature they might have published, and maybe bigger paper they might have published. But important is that what is the problem that you're thinking, whether you're bringing the solution to that or not, is a key factor, right? So then we did all the in vivo study and look at the SPARC. SPARC went down completely, significantly down, and there is on the CD31 marker, because CD31 is a marker for vessels. Means that if you put the drug should go to the tumor, means that you have to have the vessel, because it is actually going through the bloodstream, right? It has to go through the bloodstream. Drug has to go through the bloodstream to the tumor, and we want to make sure that it is the blood, blood vessels are there in the tumor area or not. If the blood, vessels are not there, then the drug cannot go in. So actually, if you see here, that CD31 is a marker for the blood vessels. So if you see, when you inhibit with the STAT3 inhibitor, actually you're seeing the blood vessels much better. Means that there is something is happening and the blood vessels are there and the tumor, that may be the reason that the drug is going in. We thought about that, right? And then we did some MRI screening and all this. Don't want to go through that. What important is that just to check the stromal density, whether it is altering the stromal density, stroma in that area. And we did some MRI study and just to make sure that it is inhibiting the stroma. But the bigger question for us at that time was whether it is remodeling the tumor microenvironment, right? The next question is the blood vessels is increased, and that means that there is something is happening to the microenvironment there, right? STAT3 we are inhibiting and affecting maybe just not the tumor, maybe surrounding around the microenvironment. Remember the way that I have done yesterday that I've showed you, like the ways that we have studied, we didn't have enough tools at that time. There is no single cell RNA sequencing. Nothing was there at that time to study. So we only look at the markers and study at that time, right? So the way we have done now is different than the way we have done during that time, it doesn't matter. The problem solved, it is the same way. We solved the problem. It doesn't matter the technology was there or not. But we were actually investigating it, and we were actually figuring it out, whether it is working, how it is working, and what exactly is happening, right? It's the same question, same thing. But it's just not, we didn't have the beautiful paper, figures like yesterday I was showing. That's all. But the result and the data is all the same. It's all the same idea, same concept and design. So this is kind of a, tumor microenvironment. Then we wanted to see whether it is normalizing the vascular and it is remodeling the stroma. These are kind of questions that we had at that time. And also we know that it reduced the tumor growth, it improved the survival, in this maybe affecting the stromal collagen. Because when it comes to stroma, there are a lot of collagen in that, because that is the reason that it will become tightened and hard. Then our, clinically, we wanted to make sure that we are disturbing the stromal area before we publish that paper. So then we went ahead, there was an investigator in Vanderbilt, she was actually working on collagen second harmonic generation imaging, not in pancreas, somewhere, some breast cancer, okay, in breast cancer field, and she was actually working with another investigator. She's actually an engineer. She actually was doing all the collagen harmonic generation imaging technology. She's completely engineer, and she was actually working with the breast cancer people to find something novel, biological difference, because remember, in those countries, what they do, even though they are engineers, they always try to find something like uniqueness in their study, and try to find some biologist or someone else, medical, or someone different than what they are working on, and try to collaborate and establish something new. That's why they were like, new technology comes, new innovative ideas comes out from those countries. The important is that collaboration and the networking and also, investigating. That's exactly the example here. Then we actually met this lady, and then we worked with her to see what is happening in our problem. Our problem is also same problem. Actually, our problem was stromal effect, and then the blood vessels is increased, the drug is going in, but what is happening to the stroma? Is it stroma is going away, or is it that the drug is really, the chemotherapy is actually affecting the stroma? What does that mean? It means that it is reducing or it's just disturbing, okay? But basically, in this study, if you see AZD1480 plus GEM, these are all the collagen fibers. It's actually, according to this data, it is actually disturbing. It is not decreasing the stroma at all. It is actually disturbing so that the vessels are going in. I told you, CD31 was actually increased. The vessels were increased. So this data is clearly proved that the vessels are disturbed. It is not the tumor going down or stroma is there. The spark data may be true, may not be true. Remember, when someone says this is the marker for this, but according to them, they found out something in their model, this is the marker. But in your model, that may be the marker, that may not be the marker. That may be something else. You never know unless you investigate and find out what is going on in your model. That's exactly what we did. We wanted to make sure the stroma reducing. If the stroma is reducing, then that means the tumor should shrink like crazy, right? It did not shrink, right? But survival increased. Everything is happening, but drug is affecting something, going there and doing something else, but we don't know what it is. That is the research. That's exactly what actually will do not give you a sleep, right? That's why you end up working so much and studying papers and come back to the laboratory, and then you're stressed and want to find out something new and what's going on. The reason is that actually the question, it's a good healthy question, is always that question in your mind, that how this is happening, right? After this study, what happened when we went back? This is exactly because I did that mass spec analysis, remember? The STAT3, SARC and EGFR inhibitor, and then chemotherapy went in. Now I wanted to see what happens to the SARC. If it is STAT3, If the target was STAT3, if I inhibit STAT3, it should happen the same way, right? That is the idea. Exactly happened. Look at what happened. So we inhibited with AstraZeneca drug 1480, that's the STAT3 inhibitor, with a gemcitabine is a chemotherapy. The drug went in. But if you see here, AZD 1480, if you just have a drug little bit, 1480 here, it is going in. It's actually going in a little bit. But together, it's going in better in this model. Right? When you have either AZD 1480 or the GEM, you see this one? AZD is also going in, gemcitabine is also going in. Don't you think this is a miracle? So SRC inhibitor did not go. Another inhibitor, so EGFR did not go. But gemcitabine went there. But here, STAT3 is also going, inhibitor, and gemcitabine is also going, that inhibitor. What does that mean? It means that STAT3 somehow is affecting some tumor microenvironment. Maybe it is something that, maybe what we found out might be unique. Maybe something that is a really signaling pathway probably is what is exactly, what the clinical trial went in, and all these patients were survived better. The reason is the drug is going in, it is affecting the STAT3 signaling. So definitely it is affecting the STAT3 signaling, that's why it's happening. Now, this data clearly confirm that STAT3 signaling, STAT3 drug is going in, that's why gemcitabine also going into the tumor. That's why those patients were survived better. We found out this, well, we were very happy, and then that paper got published in Gastroenterology, the high impact paper. The reason that went in there to the journal, the reason that because of the translatability, because what we published before, and that actually linked to this paper, and actually translatability much better, and we proved our hypothesis, what is exactly happening in that completely in that angle. So with that excitement, we were so happy, right? There is always a question. Another question will come. But as a molecular biologist, right, the question for you is this. When you inhibit STAT3, okay, everything working fine. But how do I improve better? Now the patient survived 9 months. How do I make them like in 2 years, 3 years? Right? There's that curiosity again was there. So for that curiosity, what I did, I went back to the lab, again started working on STAT3. Okay, if the STAT3 is low, what's happening? Okay, look at this data. If any of the cell line we took, these are all human pancreatic cell lines. Okay, these are all mouse lines that PANIN, PDA, LMP I showed you, liver metastatic line, and then pancreatic cancer line, and the PANIN early neoplasia line. Your STAT3 went increased, like how I showed you before, right? And the tumor went up. Look at what happened to ARC. Whenever you have high STAT3, you have a low phospho-ARC. Doesn't matter, this is a genetic though. This is the genetic, this is not a drug related. This is genetic. In the body, if the STAT3 is high, MAP kinase or the ARC is low. Okay? And you look at this one here. Whenever you have a low STAT3, there is always a high phospho-ARC, whichever cell line you take. And vice versa. If you have a low phospho-ARC, there is a high phospho-STAT3. Right? We were like, what? Why did I even worry about the phospho-ARC? Why did I go to phospho-ARC? It's not that I only went to phospho-ARC. I did a lot of other. You're only seeing the one that I did publish, right? There are a lot of work that I threw, Western blot, a lot of Western blot, like tons of it, like this much. I did not publish. The only that I published is the one that actually unique, one that I found out that is actually important to study and see what's going on. That's exactly what we got. We were like, even more interested because there was no drug at that time. Even though KRAS mutation was high in pancreatic cancer, when you have an activation of KRAS, it actually activates ERK signaling, AKT signaling. Okay? That's why this is ERK signaling is a main signaling pathway on the KRAS pathway. During that time, There was no KRAS drug. Now you have a KRAS drug. Now, at that time, we always used to write in the grant, undruggable KRAS. Now we say the undruggable become druggable KRAS signaling, right? It's a discovery from the scientists that they finally found out the drug would actually inhibit, KRAS. I'm not showing you any of those story on the KRAS in this. Anybody interested, I can discuss. But we have done a lot of KRAS inhibitor study in the laboratory. Many investigators all over the world is working on KRAS inhibitor because it's a new discovery. It's a very, very important discovery for pancreatic cancer and also other cancer, lung cancer, colon cancer. You take any cancer, KRAS is activated, right? Most of that in the colon and the lung and the pancreas. It's a very highly important signaling pathway. So at that time, there was no drug for the ERK, right, and the KRAS signaling. So, but they were all looking at the ERK signaling. ERK, we can target ERK or AKT, they were inhibiting all those. Since we got the ERK activation, we were more and more curious because KRAS signaling is important for pancreas and ERK is also important signaling pathway. Then this data took us some time, but when I got this data, wow, now my days will be spent in the laboratory again. You publish and then you continue publishing and continue thinking about what else. The curiosity is this, okay? I went to the clinical trial. After that, we published the STAT3. And now the investigation is if these patients survived better, how do I survive more? How do I make them survive more? That's the key thing. So that if you have that intention in your mind and your body, you always go back and try to investigate more and more. That curiosity you have to develop in the research. That's, I think, more important than just publication, just something. That's what I was telling you. Just publication is an important thing, but the curiosity, developing something to the patient care is also is most important thing than just the publication. So what I did after that is that, okay, I inhibited with the MEK inhibitor. These are all MEK inhibitor. This is the ERK MEK inhibitor, that is the AZD6244, or the MEK 162. They're all, so I only took the drug which are actually clinically approved drugs. What does that mean? It means that if I use the non-clinical approved drug, say for example, static here, that is a STAT3 inhibitor, well known, I think you all might know. It's the AZD1480, clinically approved. This is not clinically approved, but this drug fantastically great. But it's not clinically approved. There is some toxicity issue, there is a lot of other issues, and in vivo probably it is not able to, they are not able to use it in vivo. So we want to use a clinically approved drug. The reason is that it is easy for you to take to the clinic. Otherwise, there will be a lot of question whether this drug can be usable, whether clinically it's approvable, the toxicity level, what's happening, and a lot of these questions will come. That's why those questions might come. That's why we always try to develop clinically approved, use clinically approved drugs so that we will not have those kind of problems later on. So when we did the ERK inhibitor, look at that what happened with the time point up to 24 hours. So I'm not showing the dosage and all those things. I'm not showing all the data here. I'm just showing you, giving you a hint that it is happening, doesn't matter what. The data is clean and clear, right? So when you inhibit MEK inhibition, see what happened to STAT3, went up. So look at that MEK inhibition, STAT3 went up. And now you come here, this is STAT3 inhibitor, this is STAT3 inhibitor. You inhibit STAT3 and MEK went up. MEK-ERK is the same signaling. ERK went up, ERK went up. So what does that mean? Genetically, in the previous slide you saw, when you don't have a STAT3, you have a ERK more. Now you are inhibiting STAT3, your ERK is coming back up. And you're inhibiting ERK and STAT3 is coming back up. How fantastic it is. You know, this concept, this paper published in Cancer Research in 2018. But now if you go back and see in the field, there are many cancer, different cancer, they have the same signaling pathway. ERK is going down, STAT3 is coming up. And STAT3 going down, ERK is coming up. There are many papers. Very important. The observation that you become first, the observation is a key factor. when you observe something, and I might have thrown a lot of Western blot, a lot of things to the trash, but I selected one that is important. Probably the other person is also in the other world somewhere seeing the same thing. Probably they are looking at the same thing. They also doubt themselves sometimes whether it is true or not, right? We also doubt many times. They also doubt that whether it is true. But we model such a way, we take the genetic model, we take human cell line, you study all those things and make sure we be confident, oh, it is happening. It doesn't matter what. What I'm not showing you, different cells I screen and drug, different dosage, and all this I'm not showing you. I'm showing the data that actually clearly you can see, visualize and see it. So next curiosity for you is that what will happen if you combine MEC, ERK plus STAT3? What will happen? That's curiosity, right? So when I combine together, the both signaling pathway are going down. Means that this is a static and the U compound is a MEC inhibitor, another one. They is only work in vitro, but they don't use it for the in vivo work. I just wanted to show you just for the example, but there are a lot of in vivo drug data that we have done in the paper. But when you combine MEC and ERK inhibitor together with STAT3, the both signaling pathway are going down. There is no reactivation. They're controlling somehow. So you can see the in vivo data. These are all the pancreas tissue from the genetically engineered mouse model data that say simple Western blot data. There was no fancy way of analysis at that time. At that time when we generated, we might have published in 2018, but the data started maybe 2015, 14, 13. And it took like many years to publish, right? Because important is that publication, that work has to be impactful, means that you have to take a long time, make sure, analyze it and validate this, everything before you publish. So when you combine together, these are all going down. You know, the most important thing is for us is also EGFR, but I'll tell you the story later. But that's another story, that EGFR is also important signaling pathway, right? The SARC EGFR and the STAT3. The EGFR, there was an investigator in our Vanderbilt University. He was always asking, so did you check EGFR? Did you check EGFR? Wherever I see him, did you check EGFR? How much time will it take to check EGFR? Can you just throw EGFR antibody? And why don't you check EGFR? So it was like this, because their lab work on EGFR, they are curious to understand what's going on in our model. They always was asking, where is the EGFR? So then one day I did it, and I showed it to him. Look, I told you, it is happening. Exactly happening. Exactly what he said was happening. The EGFR was actually going down. So then he asked me, do you know why? Do you know how? I don't know. Come, I'll tell you one story. He told me one story, but that story actually became real. I'm going to show you that story. But the story he told me, but I had to have the data. I had to generate the data, that story that he is actually telling me. But it takes time for you. You have to think what the story is telling about. He's a big scientist, right? He's telling me, and then I had to go back and model it such a way that I do get the same story that what he's talking about. But to go there, that you have to really do a lot of work, right? And I went back and started working on the mechanism of understanding the whole concept. Now I know STAT3 and the MEC inhibition together is working fine. So now I think I can treat this patient much better probably if I find out how. See, in the research, you can actually go ahead and do the clinical trial like companies do. But as a researcher, you have to understand the mechanism. If you don't understand the mechanism, if you don't investigate the mechanism, either your job will go away or you'll never get the grant. Or you can go to company and join. Many people go and join the company. And probably you don't have to understand the mechanism. You can just go ahead and do the treatment and clinical trial. They'd run the clinical trial and they can do it. But as a biologist, as an investigator, understanding the mechanisms for us, how, why, these kind of questions that always should have, that's how you investigate and discover something new, right? The same question I had, what is the mechanism, how it is regulated, right? Then I was actually looking at all this that the scientists told me, the EGFR, and then I was looking at all his papers, and he was actually working on TASE, EGFR, all the signaling pathways, pro-EGFR ligand, and all these ligands, what they do, and there's kind of all these papers roaming around in their laboratory. If you talk to their graduate students, PhD students and the postdoc, they all talk about EGFR, pro-EGFR ligand, this, that. I was like, how do I study? They talk about all these things because I'm poor guy from here in the corner, one person working in the pancreas field, and with my investigator who is a clinician who actually go to OR all the time, and I was just like thinking we both together like how do we do this? And then we had to start somewhere though. So what I did was I started slowly. Started like let me look at the EGFR ligands first. Let me look at this first. Let me look at what this person is saying is true or not. Okay? The same, I went and screened all the EGFR ligands like TASE, TGF-alpha, and the amphiregulin. There was a tissue microarray that developed. Remember, the tissue microarray, probably you guys might know, it's available. There are a lot of companies sell tissue microarray of the human tissues. Like in any tissue, like in a lung, colon, pancreas, they sell it from the patients. It's available now. But during that time, we had to make our own. There was no company selling tissue microarray. So the pathologists at that time in that university, they were making the tissue microarray themselves. Because they get the tissues, they were making it. So they were actually giving us, because they are interested to know, because there are not many investigators at that time, pancreas. So we were actually took that, sorry. So we took that idea from that investigator, and we started looking at all this pro-EGFR ligand. When we saw amphiregulin, this AREG means amphiregulin, one of the EGFR ligand, actually significantly upregulated in the cancer patients. And then we look at the SARC, and then TASE is another EGFR. This is how this work. What happens is that if you go to the signaling pathway, this person published, or the other, there are a lot of other people published during that time. And even now, there are a lot of papers coming out in that area. The ADAM17, that is a TASE, actually cleaves the pro-amphiregulin, and then amphiregulin goes and binds on the EGFR. The EGFR signaling downstream, you already know, EGFR, KRAS, and ERK, AKT signaling is all downstream targeted, this signaling the whole pathway. So that is actually exactly the pathway. Even the TNF-alpha was also can be cleaved from this TASE, that is ADAM17 on the membrane. That cleaves, and then the TNF-alpha goes and activates on the other signaling pathways to the TNFR signaling pathway. So these are all kind of research that people are working on, and also their laboratory work was working on. So that's why I went back and I did all this amphiregulin. The amphiregulin actually, fortunately, we got high, significantly different. Then we looked at a lot of different things, understanding the mechanism. The amphiregulin and the SARC, TASE. During that time, there was no CRISPR. We had to do the viral transfection and then knock down all this SARC, TASE, and all those. Now it's easy. You have a CRISPR knockout, and then you can actually do it, right? So that time we would knock down, and the siRNA, all these studies we used to do, and I did that. We came up with understanding that TASE-dependent shedding of the amphiregulin, activate the EGFR. So we kind of, if you see here, we inhibited with a STAT3 inhibitor, and then STAT3 after 12 hour or 24 hour, it's actually started coming back up. Means that when you use this drug, it actually only affects for a while, and then again you have to treat, exactly like how the patients are. You take paracetamol, one day after that again you have to take, because it will come back. And the signaling also coming back, because that is the paracetamol will not work, because the drug is already gone, metabolized, and now again the fever is coming back up, and again you have to take the paracetamol. It's the same, exactly same concept. It's actually STAT3 is coming back up. Now again you have to treat with the STAT3 drug. And why it is coming back up? Because your EGFR is coming back up too. So during that time, we saw even the MEK inhibitor, ERK inhibitor is a 6244 is a ERK inhibitor. You inhibit with ERK is gone, STAT3 started coming up, and the same time EGFR is also started coming up. This is exactly what the scientist was saying. So it is through the EGFR, look at it. Probably it's something based on many factors. But exactly what happened, the EGFR was high. You inhibit STAT3, and when the STAT3 is coming back up, the EGFR is coming back up, or the vice versa, or whatever. According to what we were thinking was actually happening through the EGFR signaling. But as a scientist, you have to understand what is this signaling, the whole pathway. You don't have to go through all these things, understand this amphiregulin. But what I'm saying is that we found out that amphiregulin is actually cleaved by the TASE, and the amphiregulin going on binding on the EGFR, activated EGFR, activating STAT3, MEK, and all this signaling pathway. So that is the whole story there, right? We also combined with the EGFR inhibitor with the STAT3 and actually knocked down completely the ERK inhibitor, ERK pathway. So that means it doesn't matter whether ERK and the STAT3, even though you combine with EGFR with the STAT3, you actually can downregulate the KRAS signaling pathway. So this is very curious and also interesting data. But the whole mechanism we worked out, and that got published in Cancer Issues at that time. So, but most important problem in here is this. When you combine with STAT3 and MEK, the survival of these mice, or the tumor growth of these mice, was not so high like we thought. Like, for example, what we thought, SARC and EGFR, and the patient went and then and it was fine, survival was increased. When we did this study, the mechanism, whole thing is working. Like, how you think, how you are curious and then you want to study and you want to improve, but may not work sometime. But it's important to find out why it is not working. But this is what exactly happened. This paper would have got into even a higher impact, like more than gastro. But the problem, the paper went into cancer research, even the high impact paper, but it went into cancer research. The reason is the survival was not that great, was not improving in the mice model or in any studies that we have done, or the therapeutic study that we have done. But the whole signaling pathway, and we kind of worked out, okay? The whole mechanism we worked out. We understood the whole thing, what's going on. But the most disappointing was they did not increase so much like what we thought. But remember, there is always something, again, you're curious why it is not working. What is happening, right? So then comes this study. So we started looking at, okay, we inhibited STAT3 and MEK. Okay, we understood the whole mechanism. So it is not improving the survival. During this time, what happened was, in the whole world, in the cancer biology, tumor microenvironment, immunology concept and ideas started coming up. Everybody said, if you are not working in immunology, in cancer biology, you're wasting your time, right? Probably that's how the concept was. Even today, like if you are not doing on something immune related, it's like probably immunotherapy should be the main idea in the study in the cancer biology. Like we were like struggling to understand which immunology first, because we are cancer biologists. I had to understand and learn immunology, or we have to hire immunologists who actually can understand and tell us the story, right? So we hired the immunologist, and also same time we also learning from him. We have to understand what we did and what we did not, and he has to give me from the idea of the immunology idea, concept and ideas. That's what we learned together. And then what happened was, we screened all these tumors again, STAT3 inhibitor plus MEK inhibitor. Our interest was this. We don't want to know what happens to MEK. We don't want to know what happens to STAT3, because we already figured it out. We have to combine MEK and STAT inhibitor together. That's our first thing in our mind, okay? We don't want to know anything before. We're going to combine MEK and STAT3 inhibitor together, for sure. All we wanted to know is that how do we improve the survival? Because in pancreatic cancer, survival is a key factor. We want to make sure we increase the survival, that's it. So we don't want to know anything what happened. Now show me anywhere, any data that can improve this data further. So that's why we started with STAT3 MEK inhibition. Wherever you see MEK-I, STAT3I, that means that we are combining these two drugs and studying anything else, right? And that's what we did. We screened all the immune concept and immune cells and started working on CAF cells, immune cells, and all the population, because at that time we had the single cell sequencing day available around. And then we started looking at the single cell sequencing and then look at the different population, which population is affecting, this drug is affecting something, things like that. Because it is the drug, right, in the body, it is not only in the pancreas, it is affecting the whole microenvironment around the pancreas too. So it is not specific to only the cells are targeted, right? So it's everywhere. So it may be the drug is affecting something else is happening here. That's why we started looking at all the immune population. And in that, what happened was, if you see here, that is this is a vehicle when you add STAT3 inhibitor and the MEK inhibitor together, we started seeing the CD8 here up and the CD4 up. All this CD T cell population is going up. So that is a great thing, right? It's a great thing. But important is also that, so we looked at many different ways, like T cell trafficking in part of the CAF dependent, CAF independent, and all this. We want to know what's going on overall with this drug treatment, the combination. That's all our goal, right? But we want to look at all the cell types and we want to figure it out. That is our goal. Okay, if that is not happening with the tumor intrinsic, that should be happening to some other cell types. Something is happening. So we have to figure it out so that we can actually target this cell population. So the interesting was the PD-1. At that time, immunotherapy was coming up in the field. There were Jim Allison, who is a Nobel laureate, who actually worked on immunotherapy in MD Anderson. So they were actually publishing a paper on immunotherapy, and they even started the institute called Immunotherapy Institute in MD Anderson. So we were looking at the immunotherapy concept and ideas. And look at what happened. It is actually PD-1 actually increased when you add MEK and STAT3 inhibitor together. The PD-1, that means that we can use the PD-1 antibody to therapy, and that means immunotherapy. So we added the PD-1 antibody here. Look at what happened. The tumor is shrinking. Tumors are all gone. You look at this one. This is a completely normal tumor. Tumor is gone. So it's completely normal. So it means that, remember, there is a lot of stroma. Lot of stroma. Now I don't see any stroma. It is just, where did it go? Because some immune cells are affecting, somehow it is inhibiting stroma. Means that we were interested in CAF. If you see CAF cells, I discovered CAF cells. I mean, we were kind of working on the CAF cells and the immune cells. We were like, it's gone. Completely normal tissue. Look at the survival. Survival is so good. Look at this survival. The survival with the vehicle is a 44 days. This is a mouse model. And this is a very aggressive mouse model. This is a GEM model. That is a genetically engineered mouse model. These mice die within 8 weeks. By 4 weeks, they make a tumor. 8 weeks, they die. 8.5 weeks. Look at how many days they survived. Together, it's 181 days. It's a long time. Then we were amazed by this data. Immunotherapy together with the MEK and STAT3 inhibitor is working. And look at this one. This is I'm not kidding. This is superly good. If you see here, tumor area is nothing tumor area. The tumor area when compared to vehicle, 100%, there is hardly you see the tumor area. Okay? This is true. We were like, wow, that means that immunotherapy somehow together is working, right? And then after we treated with all three together, we looked at all the immune population. We were all fantastically tumor infiltrating T cells and cytotoxicity, functionality, We looked at all these different markers, immune-related and CAF-related markers. We looked at all those, making sure before we go ahead and do anything with this combination. And then look at what happened. We went and did the clinical therapy on the patient. We went and did the phase 1 trial and combination, because the survival unimaginable in the field, because that's like huge. So the day when we did not get the survival, even though we published, we were not satisfied. We were always thinking like something is happening. We came all the way and we got stuck here. We got stuck there for 2 years for nothing, because we were like we didn't know what to do, right? And then it came this clinical trial, and look at this patient. This is like fantastically they are doing much better. Like survival is increased, and they were like we were wondering what's happening. The PET scan, actually we don't see the tumor here, but like overall the result actually is published in gastroenterology, same last figure, because at that time there was only few patients, and this is the whole thing that we understood now is published in this paper in gastroenterology, because it went into higher paper, because the survival is increased. Survival is so good that reviewers loved it, because it's not just the reviewers loved it, it's actually happening. It's actually happening truly. Like we went and did this, the whole immune remodeling study, and it went into a phase 1 clinical trial, and then we were like, patients were fantastically doing. It's actually with combination, collaboration with many drug companies. Remember, we have a immunotherapy, and it is from the different company, and we have a MEK inhibitor, AZD, on the STAT3 inhibitor, different company. We merge these companies together. We are looking for the company who actually merge together and use those drugs together. These are all the trial problem. But what I'm saying is that you don't have to go there unless anybody is interested. So we can talk about it, how they run the trial, and that's exactly another maybe talk we can conduct and do it. And this is a combination study. Now patients are much better. Mechanistically we understood the whole concept and idea. SARC, look at the story. The story is coming from the SARC, which we were like 15 years ago. Okay, where today the patients are survived much better in the clinic. The reason is that the investigation that we have done from many years, linking one to another to another one, and from SARC to EGFR, STAT3 emerged. There is a depression. There was like patients gone, and we came back and keep doing the same thing again and again. We never stop. We're not even stopping here. Probably next time when I come, I'll tell you another story, what happened next. But that's how the research is, right? That the whole story is supposed to be, for us is a long time and important. But actually, look at the survival of the patients. Whoever is getting the drug and study, trial. Actually, now they think whatever we have done is actually done great. There are many patients gave the money for us, actually for all this work that we have done, and they loved it, because when you tell them the story of how we did all these years and how many patients survived, doesn't matter 1 month, 2 months, or 5 months, 6 months, or 9 months, or 1 year, 2 year, but whatever the time, even the one day is a great thing, right? Great feeling. So that's what I think our contribution to that field. And the future, there are a lot of things going on. Like we look at the biomarker, if this improved, what is happening in the biomarker. Many, can we combine with another, combination? Like, we did not do the chemotherapy. We did the immunotherapy, right? We want to do the chemotherapy, and together, what's going to happen, whether we can do it or not, whether it is translatable or not. This is a whole translational journey, shows how clinical, preclinical studies, like mechanistic insights, understanding the biology, then we can design this rational to a therapeutic angle, and also we can challenge. It doesn't matter what cancer. Once you learn in one cancer, you can apply the same concept, design, and everything to any cancer. But it's important for us to focus on one cancer and study the whole life. The reason I'll tell you, so that you can actually contribute so much. But the learning process that you can actually, what you learned, you can actually distribute to many other cancer areas, and probably someone will get help. What I'm doing right now, the same thing, trying to tell you that the story of this whole journey, so that you can learn something to do and conduct experiments that is actually important and meaningful to the future, right? So these are all my some collaborators. There is a Vanderbilt who actually University K. Washington is a great world renowned pathologist. The Chen Shi is another pathologist. I think Joshi knows Chen Shi. We were talking when he was there in Miami. And Nipun is another surgeon, great surgeon, pancreas surgeon, world number, probably great. He sees a lot of great patients. The main patients who are in the critical situation, critical problem. I think he sees them. So we get to know a lot of ideas from him clinically, what's going on. So that's why it's so important to collaborate with clinicians so that you get to know a lot of things. And these are all many papers that we published all these years to come, get this story and the foundation. Without money, there is no way that we can get, done any of this work, right? So money is also important. That's why the mechanistic understanding of all these things is a key factor. But at the same time, we have to have people who can actually work and establish these kind of studies. So that's the story. Thank you so much. There's any questions? I can take it. I know it took a little more time, but I don't want to stop anywhere in between. So, if you don't have any question, it's fine. We can actually discuss during the discussion time. So there is actually we have distributed the questionnaires. So in the questionnaires, there are a lot of questions. Depending on the talk that I was actually doing it. So if the mic is not working, it's not working. So it's okay, we can do it in the tutorial time because probably you will have the same questions during that time. Thank you so much. I think maybe tea time maybe? Yeah, okay, thank you.